Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to Doctor Study tutorial. In our previous video, I was discuss about the hepatitis C virus, and today I will discuss about the hepatitis D virus and E virus. Hope you enjoy this video and it will helpful you in your professional examination. So let's start. Here is the figure of hepatitis D virus. Hepatitis D virus disease. Hepatitis D or hepatitis delta, genus is delta virus, virion 35 nanometer, spherical in shape, envelope virus, genome are single stranded RNA, circular, closed circle, negative polarity, prevalence are low. It is a defective virus that acquires an hepatitis surface antigen of hepatitis B code for the transmission. It is the smallest of known human pathogen. Causes infection only in the presence of hepatitis B. Actually, hepatitis D need always hepatitis B virus for producing disease. That's why it is called defective virus, and it is a important question of viva. The RNA genome of hepatitis D is very small and encoded only one protein, the internal core protein. It is called delta antigen. That's why it is also named as delta virus. Hepatitis D has no virion polymerase. The genome RNA is replicated and transcribed by the host cell RNA polymerase. Hepatitis D virus genome RNA is an riboenzyme. It has the ability to self-cleave and self-ligate. The employed during replication of the genome. Replication cycle. Hepatitis V virus does not have the gene for its envelope protein. It cannot replicate by itself. It can replicate only in cells also infected with hepatitis B virus, because hepatitis D virus uses the surface antigen of hepatitis B as its envelope protein. Hepatitis D virus replicate in the nucleus. It is the figure of replication cycle of hepatitis D virus. Then important question is transmission and epidemiology. Hepatitis D virus infections occur worldwide with a similar distribution with that of hepatitis B infection, less prevalent in Asia. It is transmitted by the parenteral route and perinatally. Clinical findings. A person can be infected with both hepatitis D and hepatitis B virus at the same time, co-infected, or be previously infected with the hepatitis B and then super-infected with the hepatitis D. Co-infection with hepatitis D and B is more severe than the infection with the hepatitis B alone. The incidence of fulminant, life-threatening hepatitis, chronic hepatitis and liver failure is significantly higher. So we can say that the co-infection is most dangerous. Then another topic is laboratory diagnosis. Serology. Co-infection, hepatitis D antigen or hepatitis D virus RNA, anti-hepatitis D virus IgM are preferable. Superinfection, anti-hepatitis D IgM and IgG is higher. So we can here differentiate that IgM is only preferable in co-infection hepatitis D, and in super infection IgGM and IgG both are high. Supportive treatment. There are no specific antiviral therapy or vaccine against hepatitis D virus. Treatment based on controlling and treatment of hepatitis B. So this is all about hepatitis D virus.